Welcome back to the deep dive. Today we're tackling something absolutely fundamental, the engine room of metabolism, really. We're doing a critical deep dive into how we actually digest and absorb carbohydrates, proteins, and lipids. For you, our first-year MBBS listeners, getting this biochemistry down cold, well, it's the bedrock for so much in clinical medicine.
It really is. You absolutely have to grasp how the nutrients get in before you can understand, what happens next. We've really tried to synthesize this complex material from reliable sources like the standard textbooks to give you the clearest possible map. And our sources kick off with a great guiding principle kind of framing this whole massive process.
Right, that core idea. Complex is the ingested food, but digested to simpler products, absorbed by intestinal mucosal cells, assimilated and utilized by all cells. It sounds simple, but it covers everything. This isn't just abstract theory, it's literally how we function.
Exactly. So let's properly define digestion. It's not just vaguely breaking things down. It's specifically the process of hydrolysis using water chemically to split those large, complex organic macromolecules, breaking them down into much smaller, water-soluble molecules, because only those simple bits can actually get across the gut lining.
And, before the body even deploys its chemical toolkit, we give it a huge head start physically. The sources really stress how cooking, which starts denaturing proteins, gelatinizing starch, and just, chewing properly in the mouth, how much that improves overall digestibility. You're maximizing the surface area before the enzymes even get a look in.
That physical pre-processing, it's absolutely vital. Okay, so let's quickly map out the territory, the gastrointestinal toolkit. It's a highly coordinated operation.
First stop, the mouth. Here, we start some partial digestion, mainly polysaccharides, using salivary enzymes. Then the stomach introduces that intense acidity, the HCl plus acid-stable proteases. Starts on proteins, kills off bugs.
Next, the pancreas. This is your crucial chemical plant. It pumps out sodium bicarbonate, the night CO3, to neutralize all that acid coming from the stomach, protects the duodenum. And it just floods the small intestine with basically every type of digestive enzyme needed. What's really fascinating physiologically is its reserve capacity. We're talking 5 to 10 times the enzymes normally needed.
Wow, 5 to 10-fold. That seems like massive overkill. Why would the body build in that much redundancy?
It's a fantastic safety net, really, an evolutionary protection. That huge reserve means that even with, say, mild pancreatic disease, or after a really enormous meal, or even if a big chunk of the gland isn't working right, maybe up to 80% compromised, normal digestion can often still carry on fine. You only really see the clinical signs, like fatty stool, steatorrhea, when that reserve is almost completely wiped out.
Makes sense. Then you have the liver and gallbladder. They're making and storing bile acids, non-negotiable for dealing with fats. And finally, the small intestine, especially the first parts, the duodenum and jejunum. That's the main stage for the final enzyme work and for absorption itself.
And remember, this whole system isn't just about calories, about energy. The way we digest and absorb fat, specifically, is also essential for taking up those vital fat-soluble vitamins A, D, E, K. And it helps maximize absorption of certain minerals, too. It's all interconnected.
Okay, great overview. Let's unpack this bit by bit then. Starting with carbohydrates, these are the nutrients where digestion really kicks off almost the second they enter your mouth.
Absolutely. Let's understand this step by step. So, dietary carbs come mainly as polysaccharides, think starch, glycogen, also disaccharides, lactose from milk, sucrose from table sugar, and maybe small amounts of monosaccharides like glucose already present.
Right. So digestion starts right there in the mouth, thanks to salivary alpha amylase, often called TSLN. It's a hydrolase, and it specifically targets the alpha-1,4 glycosidic bonds inside the starch molecule. Chops them up randomly.
But here's a key detail you must remember for exams. It cannot break the alpha-176 glycosidic bonds. Those are the branch points in starch, like amylopectin, and it only works for a short time while you're chewing and swallowing. So what leaves the mouth isn't simple sugars, it's a mix. Maltose, maltotriose, and these crucial alpha-limit dextrins. Those are the starch fragments still containing those resistant alpha-1,6 branches.
Okay, and then that food bolus hits the stomach and boom, carb digestion just stops dead. Why?
Simple answer. pH salivary amylase is completely, irreversibly inactivated by the stomach's incredibly low pH. We're talking often below 2.0. Once it's denatured by that acid, it's gone. Even if the pH rises later, that enzyme won't work again.
So nothing more happens to carbs until the time that acidic food mixture squirts into the small intestine and meets that pancreatic bicarbonate we mentioned.
Right. That bicarbonate neutralizes the acid, brings the pH back up into the optimal range for pancreatic alpha amylase. This enzyme is very similar structurally to the salivary one. It basically picks up where the salivary amylase left off. It continues chopping away at those alpha-1 CMO4 bonds, but just like the salivary version, it still can't touch the alpha-1 and 6 bonds. So after pancreatic amylase has done its work, you've mainly got disaccharides, maltose, isomaltose, and some small oligosaccharides. Still not absorbable yet.
So the absolutely final step to get those single absorbable sugar units has to happen right at the cell surface.
Exactly. This final breakdown occurs right at the brush border, that fuzzy lining of the enterocytes, the intestinal cells, mainly in the upper jejunum. It's done by enzymes that are actually embedded in the cell membrane there.
Okay, so what are these brush border enzymes?
They're generally called oligosaccharidases and disaccharidases. You've got maltase, sucrose, and isomaltase. Together, they handle the breakdown of maltose, sucrose, isomaltose, and those small oligosaccharides into monosaccharides. But here's that really important clinical point. Lactase, which is also called beta-galactosidase. This is the specific enzyme that splits lactose, milk sugar, into glucose and galactose.
And the textbooks flag lactase specifically as being the rate-limiting enzyme for using lactose in humans. Why rate-limiting?
Well, if you measure the activity levels of all these disaccharadases in the brush border, lactase often has the lowest activity, especially after infancy, after weaning. Its activity tends to decline naturally in many populations.
And that lower activity, that rate limitation, directly links to why lactose intolerance is so common worldwide, right?
Precisely. It sets the stage for that condition.
Okay, let's move to absorption then. We've got our final products, glucose making up about 80% plus galactose and fructose. How do these single sugars actually get across the epithelial barrier, mainly happening in the duodenum and upper jejunum, you said?
Right. Now, for glucose and galactose, the mechanism is really elegant. It's a classic example of secondary active transport. They use the sodium-independent glucose transporter, or SGLT1. Remember that name? SGLT1. It's carrier-mediated, meaning it needs a protein transporter and it requires energy.
Let's break that down, because secondary active transport is a core concept you absolutely need to nail for MBBS. The cell needs to pull glucose in, often against a really steep concentration gradient. There's usually more glucose inside the cell than in the gut lumen after a meal. How does it manage that uphill battle without directly burning ATP right there at the transporter?
It cleverly uses a different energy source, the sodium gradient. See, the cell works constantly to keep the sodium ion, Ne+ concentration, low inside and high outside. This creates a strong electrochemical gradient for sodium. It really wants to get into the cell, driven by both the concentration difference and the cell's negative electrical charge inside. That gradient is maintained by the sodium-potassium ATPase pump, LHK plus ATPase, which does use ATP, but it's located elsewhere on the cell membrane, usually the basolateral side.
Ah, okay. So the SGLT1 transporter itself doesn't use ATP. It binds both sodium and glucose. And when sodium moves down, it's steep electrochemical gradient flowing into the cell.
Exactly. That downhill movement of sodium releases energy, and the SGLT1 protein harnesses that energy to simultaneously pull glucose, or galactose, they share the same transporter, into the cell, even if it's moving glucose uphill against its own concentration gradient. So the ATP use is indirect. It powers the pump that maintains the sodium gradient, which then powers the glucose uptake. It's coupled.
Okay, now pause for a moment and just think about that. It's ingenious. The body uses the energy stored in an ion gradient, like a battery maintained by a separate pump, to actively pull in essential nutrients. Brilliant energy coupling.
It really is fundamental membrane biology.
So that covers glucose and galactose. What about fructose? Is it the same complex system?
No, fructose has it easier. It uses a different transporter called GLUT5, and it gets across via facilitated diffusion. This means it still needs a carrier protein, GLUT5, but it's not dependent on sodium, and it doesn't require direct or indirect energy input. It just moves down its own concentration gradient, which is usually favorable because fructose is rapidly metabolized inside the cell. Interestingly, once inside the enterocyte, a lot of that fructose gets quickly converted into glucose, or intermediates like lactate, before it even leaves the cell. Then it exits the cell into the bloodstream, typically via another transporter called GLUT2 on the bilateral membrane, alongside glucose and galactose.
Okay, let's instantly connect this SGLT1 mechanism to clinical practice. The absolute classic example is oral rehydration therapy, ORT.
Yes, high-yield concept for sure. In things like severe cholera or other diarrheal diseases, you lose massive amounts of fluid and electrolytes. Just drinking plain water isn't very effective because the absorption mechanisms might be overwhelmed or damaged. But ORT solutions contain both sodium and glucose. Why? To exploit SGLT1. By providing glucose, you stimulate the SGLT1 transporter to pull in sodium much more effectively.
Mm.
And wherever sodium goes, water follows osmotically. So coupling sodium and glucose absorption dramatically enhances water and electrolyte uptake, essentially bypassing the damaged pathways and rehydrating the patient. Saves millions of lives.
Brilliant application. Now, on the flip side, the major abnormality for carbs, back to lactose intolerance. So it's the deficiency in that brush border enzyme, lactase. What happens to the lactose that isn't digested?
Right. If you lack sufficient lactase, lactose remains undigested in the lumen of the small intestine. Now, lactose is a disaccharide, and it's osmotically active. It holds onto water. Its presence in the gut lumen draws large amounts of water out of the intestinal cells and into the lumen by osmosis.
Causing the watery diarrhea.
Exactly. Then, if that undigested lactose reaches the large intestine, the bacteria there go wild. They can ferment lactose. This bacterial fermentation produces gases, hydrogen, H2, methane, CH4, carbon dioxide, CO2, leading to bloating and flatulence. It also produces short-chain fatty acids, which can further irritate the colon and contribute to the diarrhea and cramping. Painful stuff.
And management is mostly about avoiding lactose, right?
Primarily, yes. Severe restriction of milk and dairy products. Though, interestingly, many people with some degree of intolerance can often tolerate fermented dairy products like yogurt or curd. Why? Because the lactobacillus bacteria used in fermentation produce their own lactase, which helps predigest the lactose in the product itself. Also, in cases of acquired intolerance, say after gastroenteritis damages the brush border temporarily, just withdrawing milk for a bit might be enough for the enzyme levels to recover.
One last point on carbs and gas things, like beans causing flatulence. Is that the same mechanism?
Similar outcome, different cause. It's usually not an enzyme deficiency. Legumes contain certain oligosaccharides, like raffinose. It's a trisaccharide of galactose, glucose, and fructose. Humans simply don't have the brush border enzymes, specifically alpha-galactosidase, needed to break those particular bonds. So these undigestible oligosaccharides pass straight through to the large intestine, where, just like undigested lactose, they become food for bacteria, leading to gas production.
Okay, carbohydrates covered. Let's pivot now to proteins. You said earlier if carbs were about speed, proteins are more about safety and control. Breaking down complex proteins requires really powerful enzymes, peptidases, without, digesting ourselves.
That's the perfect way to think about it. And we classify these protein-digesting enzymes, these peptidases, based on where they cut the protein chain. You have endopeptidases. These guys attack peptide bonds within the protein chain. Examples are pepsin in the stomach and trypsin and chymotrypsin from the pancreas. They break large proteins into smaller peptides. Then you have exapeptidases. These nibble away at the ends of the protein or peptide chains. Carboxypeptidases remove amino acids one by one from the C-terminal end. Amidopeptidases remove them from the N-terminal end.
Right. And unlike carbs, there's no protein digestion starting in the mouth, correct? Saliva doesn't have proteases.
Correct. It all kicks off in the stomach
Yeah.
in that highly acidic environment.
What happens there?
Two crucial things. First, the HCl, the stomach acid, does more than just kill bacteria. It denatures dietary proteins. Think of it like unraveling a tightly wound ball of string. This exposes the internal peptide bonds, making the protein much easier for enzymes to attack. Second, the acid activates the main stomach protease. Chief cells in the stomach lining secrete an inactive precursor called pepsinogen. HCl cleaves off a small part of pepsinogen, converting it into the active enzyme, pepsin. Pepsin can also activate other pepsinogen molecules. That's autocatalysis.
And pepsin is pretty tough, right? It has to work in that acid bath.
Exactly. Pepsin is an acid-stable endopeptidase. It works best at a very low pH, around 2.0. It starts breaking down those denatured proteins into smaller polypeptides. But it doesn't completely digest them down to amino acids. It just prepares them for the next stage.
Which happens when this acidic mixture, now containing polypeptides, enters the duodenum.
Precisely. And this is where the really powerful protein digestion machinery swings into action, the pancreatic proteases. But crucially, these are secreted in an inactive form. They're secreted as zymogens or proenzymes. Think trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidases A and B.
Why inactive? Is this the safety mechanism you mentioned?
Absolutely. Critical safety mechanism. If these powerful enzymes were active inside the pancreas where they're made, they would literally start digesting the pancreas itself. That would be catastrophic pancreatitis. So they must be kept inactive until they reach the safe confines of the intestinal lumen.
Okay, so how do they get switched on only when they reach the intestine? What's the trigger?
The master switch, the key activator, is an enzyme that's not from the pancreas. It's produced by the cells lining the duodenum itself. It's called enteropeptidase, though you might still see the older name, enterokinase, sometimes.
Enteropeptidase, secreted by the duodenal cells. What does it do?
Its one incredibly specific vital job is to find trypsinogen, the inactive precursor of trypsin, and cleave off a small peptide. This cleavage converts trypsinogen into the active enzyme trypsin.
And once you have a little bit of active trypsin?
The cascade ignites. Trypsin is the key. First, it activates more trypsinogen molecules at autocatalysis again. Then, this growing pool of active trypsin turns around and activates all the other pancreatic zymogens. It converts chymotrypsinogen to chymotrypsin, proacetase to elastase, and the procarboxypeptidases to active carboxypeptidases A and B. So, enteropeptidase starts it, but trypsin amplifies and drives the whole process.
It's a very controlled chain reaction, then. What about the specificity? Do these enzymes just chop randomly?
No, highly specific. Trypsin, for example, specifically cleaves peptide bonds on the carboxyl side of basic amino acids, arginine and lysine. Chymotrypsin prefers to cleave near large hydrophobic amino acids like phenylalanine, tyrosine, and tryptophan. Elastase goes after bonds near smaller neutral residues like alanine, glycine, serine. And the carboxypeptidases A and B chew off specific amino acids from the C-terminus. Carboxypeptidase A likes aromatic or bulky aliphatic ones, while B prefers basic ones like arg and lyses. Oh, and remember, the carboxypeptidases are metalloenzymes. They need zinc, Zn2+, to function.
All that specific cutting must efficiently break down the polypeptides into much smaller pieces.
It does. The combined action results mainly in free amino acids and lots of small peptides, typically just 2 to 8 amino acids long.
But you mentioned another safety check inside the pancreas itself, just in case.
Yes, absolutely. Alongside the zymogens, the pancreatic acinar cells also secrete a small protein called pancreatic secretory trypsin inhibitor, or PSTI. You might see it called SPINK1. Its job is to be a first responder, a failsafe. If any tiny amount of trypsinogen accidentally gets activated within the pancreas or its ducts before it reaches the intestine, PSTI binds very tightly to that active trypsin and inhibits it, preventing it from triggering the full cascade prematurely. Defects in this inhibitor are actually linked to some forms of hereditary pancreatitis.
Fascinating layer of protection. Okay, so now we have free amino acids and small peptides in the small intestine lumen. How do they get absorbed?
The final digestion and absorption happen right there. The brush border comes into play again. Embedded in that membrane are aminopeptidases, which chew off amino acids from the N-terminal end of peptides, and also dipeptidases, which split dipeptides, 2 amino acids linked, into 2 free amino acids.
So the products hitting the transporters are mainly free amino acids, but also some dipeptides and tripeptides.
Exactly. The absorption isn't limited to just free amino acids. The intestinal cells can actually absorb intact dipeptides and tripeptides very efficiently.
Why would the body bother transporting small peptides? Why not just break everything down to single amino acids first?
It seems to be about efficiency and capacity. There's this specific transporter called PEPT1. It's a proton-coupled peptide transporter. PEPT1 is very high capacity and can move diamide tripeptides into the cell very rapidly, often faster than individual amino acid transporters can work. So, absorbing small peptides via PPT1 is a major route for getting amino acid nitrogen into the body.
But what happens to those peptides inside the cell? Do they enter the blood as peptides?
No, and this is a key point. Once inside the enterocyte cytoplasm, these absorbed dianetripeptides are immediately attacked by intracellular peptidases, cytosolic peptidases mainly. These enzymes rapidly hydrolyze the peptides down to their constituent free amino acids. So ultimately, what gets transported out of the enterocyte, across the basolateral membrane, and into the portal vein blood is almost exclusively free amino acids.
Got it. Peptides are absorbed for speed, but only free amino acids enter the circulation. How are those free amino acids transported? Similar to glucose?
Very similar mechanism for most amino acids. It's primarily via sodium-dependent secondary active transport, just like glucose and galactose. They use specific carrier proteins that bind both sodium and an amino acid. The energy from sodium moving down its electrochemical gradient, maintained by the Na plus Cas plus ATPase, pulls the amino acid into the cell against its gradient. There isn't just one transporter, though. There are multiple systems, at least 7 major ones, with overlapping specificities for different groups of amino acids, neutral, basic, acidic, amino acids like proline. This allows for regulation.
You mentioned an alternative pathway, too, the gamma-glutamyl cycle.
Ah, yes, the gamma-glutamyl cycle, or Meister cycle. It's another way to transport amino acids, particularly cysteine and glutamine, it seems. It involves the antioxidant molecule glutathione and the enzyme gamma-glutamyl transpeptidase. It's thought to be a rapid transport system, but it's very energetically expensive, costs about 3 ATPs per amino acid transported. So probably used in specific situations or tissues rather than being the main route for bulk absorption.
Okay, clinical connections for protein digestion. Pancreatic problems seem obvious.
Definitely. If the pancreas isn't secreting enough proteases due to chronic pancreatitis, cystic fibrosis, maybe protein digestion is severely impaired.
Mhm.
This leads to underdigested protein ending up in the feces, a condition called azoturia, and contributes to malnutrition because you just can't absorb the essential amino acids. Often happens alongside fat maldigestion, cetoria.
And the classic genetic disorder here is Hartnupp's disease.
Hartnupp's disease, yes. It's a hereditary defect. Specifically, it affects the transporter responsible for uptake of neutral amino acids in both the small intestine and the kidney tubules. We're talking amino acids like tryptophan, alanine, valine, serine, threonine,
Why is the tryptophan malabsorption the most critical aspect clinically?
because tryptophan isn't just for building proteins, it's also an essential precursor for the synthesis of niacin, which is vitamin B3. Niacin is absolutely vital because it forms the core of the coenzymes NAD+ and NADP+ AA, which are needed for hundreds of metabolic reactions, especially redox reactions. Normally, about half our niacin requirement can be met by converting tryptophan. So if you can't absorb tryptophan properly due to Hartnupp's, you effectively develop a niacin deficiency. The symptoms therefore look very much like pellagra, the classic niacin deficiency disease. The 3Ds, dermatitis, especially sun-exposed areas, diarrhea, and potentially dementia.
And how do you manage Hartnupp's?
Treatment involves giving a high-protein diet, hoping to push some neutral amino acids through other, less efficient transporters, or maybe via peptide absorption. And crucially, supplementing directly with nicotinamide, a form of niacin, to bypass the body's need to make it from tryptophan.
One last protein point. Intact protein absorption. Normally doesn't happen in adults, right?
Generally, no. The gut is designed to break proteins down completely. However, in newborns, especially in the first few days, the gut is temporarily more permeable. This allows for the absorption of intact maternal immunoglobulins, mainly IgG, from colostrum directly into the infant circulation via processes like pinocytosis or endocytosis, provides crucial passive immunity. In adults, if small amounts of intact dietary protein do leak across the gut barrier, perhaps due to inflammation or damage, this can unfortunately trigger an immune response, leading to the development of food allergies.
All right, on to the final major nutrient class, lipids, fats. And you said earlier, this is where it gets really interesting because fats are water insoluble. They need totally different handling.
Exactly. That insolubility of the watery environment of the gut lumen is the central problem to overcome. Now, there is some minor digestion that starts in the stomach. There are acid-stable lipases, one secreted by glands in the tongue, lingual lipase, swallowed with saliva, and another secreted by the stomach, gastric lipase. These work best at a slightly acidic pH and preferentially attack short-chain or medium-chain fatty acids often found in milk fat. So they are more important in infants. But in adults eating typical diets rich in long-chain triglycerides, these stomach lipases don't do much because the fat is still in large, oily globules with very little surface area accessible.
So for significant fat digestion, the absolute prerequisite step in the small intestine is emulsification. What exactly is that?
Emulsification is the process of breaking down those large dietary fat globules into much, much smaller droplets. Think microscopic. This doesn't chemically digest the fat. It just dramatically increases the total surface area of the fat that's exposed to the watery environment where the digestive enzymes work.
How is this achieved? What breaks up the fat?
It's a combination of three things. First, simple mechanical mixing from the churning action of peristalsis in the gut helps break up large globules. Second, and most importantly, the detergent action of bile salts. Remember, these are made in the liver from cholesterol, conjugated with amino acids like glycine or taurine, stored in the gallbladder and released into the duodenum. Bile salts are amphipathic. They have a hydrophobic fat-loving side and a hydrophilic water-loving side.
So they arrange themselves around the small fat droplets.
Precisely. The hydrophobic side dissolves into the surface layer of the fat droplet, while the hydrophilic, often charged, side faces outwards into the water. This coating of bile salts prevents the tiny droplets from coalescing back together into larger globules. It stabilizes the emulsion. And the third factor is that the initial products of fat digestion, things like free fatty acids and monoacylglycerols, also have some detergent surfactant properties themselves, further helping to stabilize these small emulsion droplets.
Okay, so now we have tiny bile salt-coated fat droplets. Which enzyme does the main digestive work?
The star player is pancreatic lipase. This is secreted by the pancreas in its active form, unlike the proteases. Pancreatic lipase is highly efficient. It specifically targets the ester bonds at positions 1 and 3 of the triglyceride molecule. It cleaves off the fatty acids at these outer positions. So the main products of pancreatic lipase action are 2-monoacylglycerol, or 2-MAG, where the fatty acid is still attached at position 2, and 2 molecules of free fatty acid, FFA.
But there's a complication, right? You mentioned bile salts can actually inhibit this enzyme.
Yes, it's a bit of a paradox. The bile salts needed for emulsification can actually coat the surface of the emulsion droplets so effectively that they block pancreatic lipase from getting access to the triglycerides inside. The solution is another pancreatic secretion, colipase.
Colipase, what's its role?
Colipase is secreted as an inactive procolipase and gets activated by trypsin in the intestine. Active colipase essentially acts as an anchor. It binds both to the bile salt-coated surface of the lipid droplet and to pancreatic lipase itself. It basically displaces some bile salts and holds the lipase firmly at the oil-water interface, allowing the lipase to overcome the inhibition and efficiently hydrolyze the triglycerides. So colipase is an essential cofactor for pancreatic lipase function in the presence of bile salts. You need both.
Okay, we also digest other lipids besides triglycerides, right?
Yes. The pancreas also secretes cholesterol estrase, which hydrolyzes cholesterol esters, cholesterol linked to a fatty acid, into free cholesterol and a fatty acid. And phospholipase A2, which is secreted as a proenzyme and activated by trypsin. It cleaves the fatty acid from position 2 of phospholipids, like phosphatidylcholine and lecithin from bile, producing a free fatty acid and a lysophospholipid.
So now we have a mixture of 2-MAG, free fatty acids, long chain mostly, free cholesterol, lysophospholipids, and fat-soluble vitamins in the gut lumen. Still insoluble. How do they get absorbed? This seems like the biggest hurdle.
This is where mixed micelles come in. This is different from the emulsion droplets. Micelles are much smaller aggregates formed spontaneously by bile salts once their concentration reaches a certain level, the critical micellar concentration. Bile salts arrange themselves into tiny disc-like structures. The hydrophobic parts face inward, creating a tiny hydrophobic core, and the hydrophilic parts face outward into the water. The digestion products, the 2-MAG, long-chain FFAs, cholesterol, lysophospholipids, fat-soluble vitamins, partition into the hydrophobic core of these mixed micelles.
So the micelles act like tiny transport shuttles.
Exactly. They are soluble in the watery environment of the gut lumen because of the outward-facing hydrophilic parts of the bile salts. They can diffuse through the unstirred water layer, a layer of relatively stagnant fluid right next to the surface of the intestinal cells, which is a barrier for free lipids. The micelles ferry their lipid cargo right up to the brush border membrane of the enterocyte.
And then what happens? Does the whole micelle get absorbed?
No, the current thinking is that the lipids simply diffuse out of the micelle and directly across the lipid bilayer of the enterocyte membrane, moving down their concentration gradient, which is kept low inside the cell as they are quickly processed. The bile salts themselves are mostly left behind in the lumen. They release their cargo, pick up more lipids, and repeat the process. Most bile salts are actually reabsorbed much further down in the terminal ileum to be recycled by the liver. That's the enterohepatic circulation.
Is micelle formation needed for all fatty acids?
No, key exception. Short-chain and medium-chain fatty acids, generally less than 10 or 12 carbons long. These are sufficiently water-soluble that they don't really need micelles. They can be absorbed directly across the enterocyte membrane, probably by diffusion.
Okay, so the lipids are inside the enterocyte now. What happens next? It's different for short, medium versus long chains, you said.
Totally different fates. The short and medium-chain fatty acids, SMCFAs, have a simple journey. They are not reesterified back into triglycerides inside the cell. They pass straight through the enterocyte, exit across the basolateral membrane, and enter the portal blood directly. In the blood, they bind to the protein albumin and are carried straight to the liver for metabolism.
But the long-chain fatty acids, LCFAs, the 2-MAG, the cholesterol, they take a much more complex route.
Yes, the long-chain route is unique. Inside the enterocyte, primarily in the smooth endoplasmic reticulum, these components are reassembled. First, the LCFAs must be activated. Synthetase, or thiokinase, to attach them to coenzyme A, forming fatty acyl-CoA. Then, these activated LCFAs are reesterified back onto the 2-monoacylglycerol backbone to reform triacylglycerols, TAGs. Cholesterol is also reesterified to cholesterol esters.
Why rebuild the triglycerides? We just broke them down.
It seems counterintuitive, but it's about packaging for transport. These reformed TAGs, along with cholesterol esters and fat-soluble vitamins, are extremely hydrophobic. They can't just dissolve in the blood. So inside the enterocyte, ER and Golgi, they get packaged into large lipoprotein particles called chylomicrons. This packaging involves coating the hydrophobic lipid core with more amphipathic molecules, phospholipids, free cholesterol, and specific proteins called apolipoprotein.
Which apolipoprotein is key here?
The main structural one assembled into chylomicrons in the gut is apolipoprotein B48, ApoB48. It's essential for forming and secreting the chylomicron particle. You also get others like ApoAI added.
Okay, so we have these big balls of reassembled fat coated with proteins. How do they get out of the cell and where do they go? Not the portal vein, you said.
Correct. Chylomicrons are too large to enter the blood capillaries directly. They are released from the enterocyte by exocytosis at the basolateral membrane, and they enter the lymphatic capillaries, called lacteals, which are present in the core of each intestinal villus and have larger openings than blood capillaries. The entry of these fat-rich chylomicrons into the lymph gives it a milky appearance after a fatty meal, hence the name lacteal from Latin lac, milk.
So they travel through the lymphatic system first?
Yes, the lymph fluid eventually collects into larger lymphatic vessels, flows up through the thoracic duct, and finally empties into the bloodstream, typically near the left subclavian vein.
Why this complicated lymphatic detour? Why not just dump the fat straight into the portal vein like sugars and amino acids?
The key reason is thought to be protecting the liver from being suddenly overwhelmed by a massive flood of dietary fat after a meal. By entering the systemic circulation gradually via the lymph, the chylomicrons can first circulate to peripheral tissues, like adipose tissue for storage, and muscle for energy. Enzymes on the capillaries in these tissues, lipoprotein lipase, can break down the triglycerides in the chylomicrons, allowing those tissues to take up the fatty acids before the chylomicron remnants eventually reach the liver. It distributes the load.
Amazing system. Clinically, if any part of this fat digestion absorption pathway fails, the result is steatorrhea.
Yes, steatorrhea, fatty, bulky, foul-smelling stools is the hallmark of fat malabsorption. It can be caused by problems with digestion, lack of bile salts due to biliary obstruction or severe liver disease, lack of pancreatic lipase chylipase due to chronic pancreatitis or cystic fibrosis, or problems with absorption itself, damage to the intestinal mucosa like in celiac disease, or rare defects in chylomicron formation secretion.
And this pathway is a target for anti-obesity drugs.
Definitely. Orlistat, Xenical, Ali, is a widely used drug. It's a synthetic molecule that covalently binds to and inhibits pancreatic lipase and gastric lipase too. By blocking the main fat-digesting enzyme, it prevents the breakdown of dietary triglycerides, meaning they can't be absorbed. About 30% of ingested fat passes through undigested and is excreted. Another approach was Olestra. This is a synthetic fat substitute made by esterifying sucrose with fatty acids. It tastes like fat, but the linkages are such that human lipases cannot hydrolyze it at all. So it passes through undigested, providing no calories, but could cause unpleasant GI side effects and loss of fat-soluble vitamins.
Nearly there. Before we summarize, a quick word on digesting the nucleic acids in our food DNA and RNA.
Right, we do eat cells, so we ingest nucleic acids. Their digestion happens mainly in the small intestine, thanks again to pancreatic enzymes. The pancreas secretes ribonucleases, RNases, and deoxyribonucleases, DNases, which break down RNA and DNA into shorter chains called oligonucleotides.
And then further breakdown.
Yes. Intestinal enzymes, called phosphodiesterases, then cleave the oligonucleotides into individual mononucleotides. Finally, enzymes called nucleotidases remove the phosphate group, leaving nucleosides, base plus sugar, and then nucleosidase can split the base from the sugar. The resulting free bases, purines, pyrimidines, pentose sugars, and phosphate, are then absorbed by the intestinal cells, often via specific transporters. They can be reused by the body, salvage pathways, or further metabolized and excreted, like purines ending up as uric acid.
Got it. Now let's tie everything together by quickly revisiting some key clinical conditions involving digestion absorption failures.
Okay. First, peptic ulcers. We mentioned pepsin and HCl in the stomach. Ulcers are essentially breaches in the protected mucosal lining of the stomach or duodenum caused by damage exceeding repair. Historically thought to be mainly due to stress and excess acid, but the huge discovery was the role of the bacterium Helicobacter pylori.
How does H. pylori contribute?
It colonizes the stomach lining, causing chronic inflammation, gastritis. This inflammation weakens the mucosal defenses, the mucus layer, the bicarbonate secretion, the tight junctions between cells. This makes the underlying tissue much more vulnerable to damage from the normal levels of acid and pepsin, leading to ulcer formation. So modern management must include antibiotics to eradicate H. pylori, often combined with drugs to suppress acid, like proton pump inhibitors, to allow healing.
Second, pancreatitis, inflammation of the pancreas. You mentioned the dangers.
Yes, acute pancreatitis can be life-threatening, often triggered by gallstones blocking the pancreatic duct or by heavy alcohol use. The key event is the inappropriate, premature activation of pancreatic zymogens, especially trypsinogen to trypsin, within the pancreas itself. This active trypsin then triggers the whole cascade, leading to autodigestion of the pancreatic tissue, inflammation, hemorrhage, necrosis. It's diagnosed clinically by severe abdominal pain and markedly elevated serum levels of amylase and lipase leaking from the damaged cells. Chronic pancreatitis, often linked to long-term alcohol abuse, involves progressive inflammation, fibrosis, and destruction of pancreatic tissue, eventually leading to loss of both exocrine, digestive enzymes, and endocrine insulin function. Results in maldigestion and often diabetes.
And third, celiac disease, also called celiac sprue or gluten-sensitive enteropathy.
A classic malabsorption syndrome. It's an immune-mediated condition triggered by dietary gluten, found in wheat, barley, rye, in genetically susceptible individuals. Specifically, a component of gluten called gliadin initiates an inflammatory response in the small intestine. This immune attack damages and flattens the intestinal villi that's called villus atrophy. Losing the villi drastically reduces the surface area available for nutrient absorption and also damages the brush border, impairing enzyme function. Leads to malabsorption of virtually all nutrients, fats, carbs, proteins, vitamins, minerals.
And the treatment is absolute?
Yes. The only effective treatment is a strict, lifelong gluten-free diet. Removing the trigger allows the intestinal lining to heal and absorption to return to normal. Requires careful dietary management.
Yeah.
Okay, that was a comprehensive run-through. From basic hydrolysis to transport mechanisms right into key clinical scenarios. Let's try and crystallize the absolute must-know points.
Right. Focus your memory on those three fundamentally different transport strategies dictated by what's being absorbed.
1. That's sodium-dependent secondary active transport. Remember SGLT1 for glucose and galactose, and similar Na plus coupled systems for most L-amino acids. Energy comes indirectly from the Na plus K plus pump maintaining the sodium gradient.
2. Facilitated diffusion. Simpler, Na plus independent. Key example is GLUT5 for fructose. Moves down its own gradient.
And 3. The unique lipid pathway. Mixed micelle formation using bile salts to shuttle lipids to the cell surface, followed by diffusion in, then intracellular reesterification of long-chain components, packaging into chylomicrons, needing ApoB48, and mandatory exit via the lymphatics.
And for proteins, don't forget the safety aspect. The whole pancreatic zymogen cascade is kicked off only by enteropeptidase on the duodenal brush border. Trypsin then runs the show, but PSTI, SPION-K1, acts as the internal bodyguard within the pancreas.
And clinically, the single most common defect you'll see related to digestion is lactose intolerance. Directly links the missing enzyme, lactase, to the symptoms, osmotic diarrhea, gas from bacterial fermentation, via basic physiology.
Okay, let's lock this in with a couple of quick recall questions for you.
Question 1. Lipids. Think about that complex fat absorption. Besides pancreatic lipase itself, what two other substances are absolutely essential for efficiently digesting and then absorbing long-chain triglycerides? And what specific apolipoprotein is non-negotiable for getting those absorbed fats out of the intestinal cell?
Give you a second. Pause. Got it. The answers are bile salts for emulsification and micelle formation, and collapase to help lipase work with bile salts. And the essential protein for export is apolipoprotein B48 for chylomicron assembly.
Excellent. Question 2. Protein clinical. Remember Hartnupp's disease. It causes poor absorption of neutral amino acids, especially tryptophan. Why does this specific amino acid defect lead to symptoms resembling a particular vitamin deficiency? And what supplement is key in management?
Think about tryptophan's other roles.
Plus.
Right. Tryptophan is needed to synthesize niacin, vitamin B3, NAD+. So the malabsorption mimics pellagra. Treatment requires nicotinamide supplementation.
Fantastic. If you got those, you're really integrating the biochemistry with the clinical relevance. Great job. Yeah. You've genuinely mastered another key foundation block in biochemistry. Understanding why these processes work and how they fail is what makes you more than just a memorizer. It makes you a thinking clinician.
Absolutely. And maybe one final thought to leave you with. We talked about orlistat blocking pancreatic lipase, and we know the pancreas has this huge 5 to 10-fold reserve capacity. So thinking ahead to pharmacology and real patients, If someone is on a drug like orlistat long-term, how might the body try to adapt or compensate for that blockade, given its inherent redundancy? Could effectiveness change over time? Something to mull over.
Definitely food for thought. We'll
Right.
catch you on the next deep dive.